Bismillahirrahmanirrahim. Assalamu alaikum. In the previous lecture, we were discussing the RBC, how they are formed. Regarding that, I asked you one question. What is the size of the nucleus of mature RBC? The answer to this question is that mature RBC has no nucleus to give more space to the hemoglobin, so that without nucleus, we gain more space in the RBC to be attached for the attachment to the hemoglobin molecules. So, the next question is, if it has no nucleus, then how it maintains its life? I am telling you these questions because they are usually asked in the Viva. So you must know the answer to these Viva questions, and they are not written as such in Guyton. So remember them clearly and also copy the answer of these questions. In the mature RBC that contains enzymes required for glycogen metabolism, the balance between synthesis and utilization is such that no significant amount of glycogen accumulates within the cell under normal circumstances. Glycogen may accumulate, however, in glycogen storage diseases, lacking a storage compound, the normal erythrocyte must have constant access to glucose if its energy metabolism is to be sustained. In short, under normal circumstances, about 90% of the glucose entering the red cell is metabolized by the anaerobic pathway and 10% by the aerobic pathway. So that's the reason behind the existence of RBC without nucleus. Now coming to the new topic, that is the formation of hemoglobin. How it is formed. Synthesis of hemoglobin begins in the proerythroblast and continues even into the reticulocyte stage. When reticulocytes leave the bone marrow and come to the bloodstream, they continue to form minute quantities of hemoglobin for another day or so until they become mature erythrocytes. So, early in the proerythroblast, the formation of hemoglobin starts and continues after its reticulocyte stage. The steps involved in the formation of hemoglobin includes that succinyl-CoA combines with glycine to form pyrrole ring. 4 pyrrole rings they combine to form the protoporphyrin, which combines with iron to form the heme. It combines with the globulin molecule present in the plasma, which is simply the plasma protein, to form the hemoglobin chain. And what is hemoglobin chain? That is actually 2 alpha and 2 beta chains which forms the adult hemoglobin. Now I am saying the adult hemoglobin. Why I am using this word? Because we have different types of the hemoglobin molecules at different developmental stages. In this picture you can see the alpha chains and the beta chains. This is the basic structure of the hemoglobin molecule with the attachment of the iron and heme group. And I want to show you another animation, very nice animation, to show the 4 hemoglobin subunits alpha and beta chains combined with the 1 oxygen molecule. Hemoglobin consists of protein subunits of amino acids called polypeptides. At this stage, I must tell you that today's lectures material is from different books. It is not written as such in Guyton, so you must concentrate each and every slide and remember them clearly. In this lecture, I am going to tell you how the adult hemoglobin forms. Adult hemoglobin means that in neonates, during our fetal life, we have a different type of the hemoglobin, while with the age, this type of the hemoglobin changes. How it occurs, that is the question. In the embryo, we have different types of the hemoglobin, GAU1 and 2, and hemoglobin Portland. In the fetus, we have fetal hemoglobin, and in the adult, we have adult hemoglobin. Where lies the difference? The difference is between the chains of the hemoglobin. In the embryo, at that stage, we have different types of the hemoglobin chain. In the fetus and in the adult, we have different types of the chains. For the first 3 months, we get embryonic hemoglobin, which comes as one of the 3 types, GAU1, GAU2 or Portland. Then for the rest of the pregnancy, we get fetal hemoglobin. At which age this embryonic hemoglobin changes to fetal hemoglobin and fetal hemoglobin changes to adult hemoglobin. This is a very important viva question, and the answer to this question is this one, this graph. In the upper portion of this graph, I am showing you the areas for the erythropoiesis. This is the period that is the prenatal and this is the postnatal. This demarcation is birth. Before birth and after birth, these are the areas for erythropoiesis. In the lower portion, I am going to tell you the different types of the hemoglobin and which age the different types that changes, embryonic changes to fetal and fetal changes to adult. You can see at 3 months of the prenatal age, almost embryonic hemoglobin that is changed to the fetal hemoglobin, and after birth almost at the 1.5 month or 2 months interchange occur, and almost postnatal 6 month the fetal hemoglobin that is completely changed by adult hemoglobin. The most important function of the hemoglobin is its combination or its transport for the oxygen. It carries oxygen from the lungs and drop it to the tissues to be supplied at cellular level. For that one, it combines with the oxygen, it picks oxygen from the lungs and drop it to the tissue level, from where it picks the carbon dioxide and take that one to the lungs to be exhaled out. The detail of this will be discussed later on, but this combination of the oxygen with the hemoglobin is called the oxyhemoglobin, and how it dissociates or it gives oxygen to the tissues that is shown in this graph or curve. This is the lung level. At the lung level, hemoglobin combines with the oxygen and take it to the tissue level here, at which it delivers oxygen. This is a very specific graph or combination of the oxygen and hemoglobin. The details of this graph that will be discussed later on while we will discuss the respiratory unit, in which I will tell you the details of oxyhemoglobin combination and its dissociation at tissue level, and how the carbon dioxide replaces oxygen and combines with the hemoglobin to come to the lung level, and from where from the lungs it is exhaled. At this stage, you must remember that the main function of the hemoglobin is to transport oxygen, to pick the oxygen from the lung level and to drop it to the tissue level. As I have told you already, that for the formation of hemoglobin, we require iron, which is very important to form the heme portion of the hemoglobin. So we must know what is the daily requirement of the iron, how it is absorbed, transported and utilized in our body. It is absorbed in proximal duodenum, and in diet it is mainly present in ferric form, and on brush border it is converted into ferrous form. What is meant by brush border? Which is inside towards the bolus of the food. So from there, brush border, because it contains many villi to increase its surface area. From there, the iron is absorbed, then enters into enterocytes. Enterocytes means the intestinal cells. From there, it is absorbed. It can be stored as ferritin or transported across basolateral membrane by ferroportin 1 transporter. Basolateral membrane is the specific membrane which is outer intestinal membrane. So inner intestinal membrane is the brush border and outer is basolateral membrane. Further, it combines with the beta globulin apoferritin to form transferrin. One transferrin can bind two molecules of the iron. This iron can be released to any tissue cell at any point in the body, especially to bone marrows and the liver. In short, you can see this one. From the food, we are taking the iron. It may combine with the heme to form the heme molecule. Iron is going to be attached with there to form the heme molecule, and this is the non-heme iron. Then it is transported to the brush border, from where it is coming inside the enterocytes. In short, this is the story. In small intestine, iron is combined with the apotransferrin to form the transferrin, transferred and transported in plasma as transferrin, and in the cell cytoplasm, iron is combined with apoferrin to form the ferrin. Again, this is a very nice picture to show how we are taking iron, how it is absorbed, stored, and what is the further fate. Lifespan of the RBC is 120 days, after which they are destroyed. Regarding the hemoglobin, we have two things, heme and globin. Globin is just simple plasma protein, while heme, in that we have the iron that is carried by transferrin to the bone marrow, and it may be stored as ferritin, and porphyrin ring is converted to bilirubin. This is the heme catabolism. You can see from the RBC we are getting hemoglobin, then globin is plasma protein, heme is coming to the liver and to be excreted out through the kidney and in the through the intestine. Now lastly we have the question of the day. How many molecules of hemoglobin are attached to one RBC? So this is your assignment also, and it is a very important Viva question. Kindly search the answer, and we will start the next lecture with the answer of this question. Okay, thanks and Allah Hafiz.